Hello, sir. Presentation for pre-PhD coursework seminar. My title name is Biodegradation of Industrially Important Textile Dyes by Actinomycetes. My name is Prashik Warbhuvan. Under the guidance of my PhD, Dr. Narsinghe AP. Let's start the introduction. Azo dyes are in textile industries. These are useless after are left in different water bodies such as rivers, lakes, etc. These dyes are not easily degradable in the environment and cause carcinogenic effect, mutagenic effects, toxic to the aquatic life. The textile industry azo dyes are used to dyeing purpose on large scale due to it's comfortable to use in production in different variety in colorful compound cloths. Actually, this topic choice behind the some reason. When I was working for a pharmaceutical company, the atmosphere near to them was very bad and get a different kind of smell as well as that is the reason I wanted to do something on prevention of pollution. We have very big textile industry uh, in our country and dyes are used to make colorful cloths which contribute to the pollution. The environmental pollution caused by the release of industrial wastewater contaminated with a wide range of azo dyes is a serious problem in present days. Azo compound constitute the largest and the most diverse group of synthetic dyes that are widely used in the number of industries such as textile, food, cosmetic, paper printing. Azo dyes containing one or more azo bond minus n is equal to n minus account of for 60 to 70% all textile dye stuff used. These dyes are recarbonated to biodegradation due to the addition to the toxicity. Let's talk about the problems. Environmental pollution has been recognized as one of the major hazard of the modern world. The dyes are produced from known carcinogenic like benzodiazepine. These are directly extracted into the water bodies without treatment, which cause harm to the aquatic ecosystem. Textile dyeing wastewater is one of the most important source of pollution. Other dyes are highly water soluble in nature. High concentration of dyes in water cuts off sunlight penetration and it causes upset biological activity of the aquatic system. The dyes are produced from known carcinogenic mutagenic agent are drained into water bodies without treatment. My idea to decolorize the azo dye. Acromycites can be used to decolorize the azo dyes, which are factor polluting water and land. Economic and safe removal of the polluting dyes is still an important issue. I am focusing on short time duration for decolorization and degradation. Let's talk about the actinomycete. Gram-positive bacteria commonly found in soil. Colonies of the actinomycetes from fungus-like branches, not easy to culture and isolate. Actinomycetes usually 1 to 2 diameter. Actinomycetes are microorganisms which can completely decolorize azo dyes. The process of decolorization can be supplemented with the different carbon and nitrogen sources to enhance the degradation of dyes. My objectives are collection of samples, enrichment of dye degradation actinomycetes, isolation of dye degrading actinomycetes, identification of dye degrading actinomycetes. My working plan is collection of literature articles from internet and other sources. Collection of dyes. I'm also collected dyes from various textile industries. That dye name was yellow C4G, navy blue BNC, yellow M4G, red M5B, reactive orange M2R from various industries. Also, I'm collecting water samples from Amit process, Veerbhadra process, Vishnu process, Anand process, Raghunandan process, textile industries, Ichalkaranji on 2nd and 3rd January 2023. Here are some pictures. I'm collecting water samples, soil sample, dye samples. These are the textile industry drainage. They are directly drain the water into drainage, rivers, lakes. The use to the in last 2 years participate in the poster presentation in Goa as part of the international conference on environmental sustainability and the biotechnology 2022 opportunities and challenges at Goa. RUSA sponsored international conference on emerging trend in applied microbiology and food science 2022 at Satara. Here's some picture of poster presentation in Satara. Also the pictures poster presentation in Goa, interaction with the conference head. My methodology is the study conducted at the Department of KSK College, Bid. I'm using the culture media, mineral salt medium, starch casing agar, cytometric isolation agar. Also the sample correction is done. Contaminated water samples and effluent or contaminated soil we are collected from Amit process, Veerbarda process, Vishnu process, Anand process, Raghunandan process, textile industry in Isalkaranji, India. These samples were used to source to isolate the bacteria with the distinct morphological colony characteristic capable of the decolorizing selected azodie. The contaminated water and effluent samples were collected in airtight bottle and filtered through ordinary filter paper to remove the large suspended particles, and the filter were used to the isolation of actinomycetes. Yellow C4G, navy blue BNC, yellow M4G, red M5B, reactive orange M2R dye was collected from textile industry Karanji. All chemicals used were are highest purity and the analytical grade. I'm using the enrichment and isolation of dye-degrading actinomycetes. The actinomycetes will be isolated from samples collected waste disposal site of the Veerbhadra process, Vishnu process, Anand process, Raghunandan process, Karanji, India, by the enrichment culture technique. The sample collected will be subjected to the enrichment culture technique. The enrichment will be carried out in 100 ml mineral salt medium containing 0.1% LOC4G dye taken in 250 ml enrichment flask by adding the 10 ml of each sample separately. The flask will be incubated in rotary shaker at 50 RPM at the 30° for 1 week. Isolation and characterization of dye degradation actinomycetes. A luful culture from the enrichment flask was streaked on sterile mineral salt medium agar plates or actinomycete isolation agar plates or starch casing agar plates with 0.1% LOC4G and was incubated at room temperature for 1 week. Similarly, effluent sample will directly streaked on sterile MSM agar plates with 0.01% of LOC4G dye and incubated at room temperature for 1 week. The identification of dye degradation actinomycetes strains will be carried out based on morphological, culture, and physiological characteristics of colonies obtained on the plates. My observation and result: MSM, mineral salt medium agar plates containing 0.1% of LOC4G dye will be observed for the growth of actinomycetes and dye degrading potential. The isolate organism which have the ability to degrade the dye will be identified based on morphological, culture, and physiological characteristic. Here's some my methodology. I'm also attending science exhibition at KSK College 2023. Here's some my actinomyces isolated from the textile industry soil samples. Targeted outcomes. The society and the targeted outcomes for my research is prevention of carcinogenic components, prevention of mutagenic components, prevention of toxic chemicals to the water bodies, prevention of land pollution. Acknowledgement. I would like to thank to the Dr. Anil Narsinghe sir for his continuous guidance and support for my research work. He gave me the inspiration to do project on the biodegradation. He really helped me to understand the importance of collection of samples property in the isolated way. I would like also thank to the Dr. Gurve Madam and my research center, KSK College Bid. Here some my references related my research topic. Okay.
Okay.
Thank you, sir.
Nice presentation by Prashik Warbhuvan. Seminar presentation on the topic biodegradation of textile dyes by the actinomycetes. Thank you for your nice presentation. Thank you.
Thank you, sir. Thank you.